Hello, everyone, and thank you for being here with us on our episode 7 holiday edition. This is our episode 7 before the end of the year, and we are very excited to wrap up the year. This year has been challenging and arduous in reaching out to the research community after 2 years of COVID. So, with that in mind, we want to wrap up the year with some summaries of what we've learned from this year, what COVID left us, what we look forward to next year, hopefully bettering of meetings. We know that some people have already done in-person meetings. There are some still things going on, but we know that we are very excited for things moving forward. So with that in mind, maybe you guys want to share, Daisy, Vic, do you guys have something in mind?
Yeah, so I'm actually really excited that we launched this podcast this year and managed to push out 7 episodes, which is fantastic. And thanks everyone for watching and listening. And thanks to all our guests for coming on board and also taking selfies with their BehindOurScience mugs, which has been super awesome to see. Well, we have something very exciting coming up for next year. Lots of great things in the works. One of which is actually collaboration with a really big company. It's our first kind of collaborative sponsorship from an industry partner. So that's kind of fun. And so yeah, BehindOurScience is moving on up. And yeah, you'll hear more about it. But hint that a Greek letter may be involved in this company. And we will be highlighting some really exciting people who have accomplished what we deem to be seemingly impossible things, and they've accomplished so much in science, and we want to highlight and share their stories behind the science.
I guess what I've learned from this year is that you're able to get through whatever is put in front of you. So I recently defended my PhD dissertation, and I really thought I would never reach that point, but I reached it and it's done. And it was hybrid format, which was really weird. And I know we've had a taste of that with conferences, but whenever you're trying to prove that your science is worth it to the experts in your institution, it's a little daunting. But I would say that this COVID, not season because it's been years, the COVID years have taught me that we're way more resilient than we initially thought. And one of the things that I'm excited about for this podcast is that Emily Essex, who's the managing editor of the American Journal of Pathology, will be discussing future directions and highlights for their journal. In fact, we have her clip for this episode, so that's really exciting. And I hope that the listeners get to enjoy that in the future.
Overall, the greatest accomplishment, and I'm going to try to make this a trend where we actually say BC and it means before COVID and after COVID, so that people relate to all of these terms moving forward. I think COVID hit us in the soft spot. It showed us how vulnerable we are, and I agree with Vic about being resilient. We actually don't know how difficult this will be, but I guess moving forward, we'll be able to tell our grandchildren, I was part of it and we made it. We made through it. So with that in mind, I think that's what we've learned. We've learned that many things have been accomplished. I actually want to thank Vic, Daisy, and Marina, Marina couldn't be here because there's a couple of inclement weather going on in Greece right now. We have a couple of clips from her. But excitingly, I know that this year has been a struggle. I proposed this weird and crazy idea to Daisy, and then we got Marina involved, and then we got Vic involved, and now we've become a group that identifies with all of these different efforts in doing scientific communication. And I know that we've struggled because we want to keep our science going, with COVID, through COVID, with interviews, with thesis, with everything going on. But we make the episodes thinking about you guys, thinking about how to make it better, how to make science communication available for people, because we know it's hard. So with that, we'd like to thank the American Society of Investigative Pathology. They completely supported us, have been amazing. Bill Coleman, Lisa, Gina, everyone has been amazing in doing this, as well as the Histochemical Society, Kendra, Alex, everyone in the society. We've been getting more and more involved with them, and I believe there's interesting things happening. We have been trying to reach into a company called Upper Deck, which they make all of these memorabilia trading cards, all of these Sports Illustrated, Sport theme cards. So we have an initiative to actually make those cards highlighting our membership. We've given out mugs and stuff in the past as memorabilia of being in the show or being part of the episodes or anything related to the podcast. And now we want to feature people that they have something to demonstrate how amazing they are and how enthusiastic we are with having them join our podcast. So with that in mind, we're going to have many things to come in the following year. The podcast is continuing. We still have support from our sponsors and from people that want to continue watching this podcast.
Hello, Behind Our Science folks. I wanted to jump in and wish you all happy holidays. Wishing with the rest of the guys because of some weather circumstances. Aristos, please take the time to care for your mental health. Be kind to one another. Spread love and positivity. Hear each other out. Be compassionate scientists. Prioritize you. Last thing, remember to check out your favorite science journals. They could have editorials that you're really interested in. I highly recommend the Histochemical Society's journal and the American Society for Investigative Pathology's journal. But hey, I'm biased. We have many things in the works for you, including interviews, impressive scientists that are working, train the next generation of scientists like Jennifer Sanders, as well as scientists that make a difference in classrooms worldwide, like Sarah from Skype a Scientist. We also have a fun segment called Science Siblings that is coming up. Angelic sisters were the first one to be interviewed. There's many more, so stay tuned. During this time off, please take the time to rest. You know that your everyday actions are aligned with your values. Make sure you connect with the people you love and trust a little voice in your head that tells you to follow your bliss. No matter how hard it is, it's gonna be okay. Happy holidays and have fun. Be a responsible driver. Don't eat too much all the time. Wear a mask. Bye.
Wishing you all happy holidays from the lab.
Keep your pipettes warm for the holidays with beanies.
My favorite Christmas thing in the lab is a centrifuge.
Alright, my Christmas item in the lab is the iBlock 2 machine for dry transfers in the Western blot.
My favorite thing in lab? This guy.
My favorite Christmas thing is the HPLC.
Hey, so I'm Said and this is my favorite thing, my EBOS microscope.
Okay, my favorite Christmas thing in this lab is BT1000 pipette.
My favorite Christmas thing in the lab is the liquid nitrogen tank.
My favorite thing in the lab is the dissection microscope.
So my favorite thing in the lab is the axioscope.
My favorite Christmas thing in the lab is the ethanol spray bottle.
My favorite Christmas thing in the lab is Daisy's Pipette Boy. My favorite Christmas thing in the lab is this calcium machine with the cute little hat decoration on.
My favorite Christmas thing in the lab is our microcentrifuge.
Before you go, Emily Essex here with your Journal Minute. The December issue of AJP is now available, and it contains 2 new review articles, both of which are fully free to access for all readers, no subscription required. One of them focuses on the pathogenesis of osteochondroma, and the second is on lymphangiogenesis and lymph node metastasis in liver cancer. Both of these review articles have ASIP member authors, and we hope that you will go check them out online at ajp.amjpathol.org. See you next time.
This month's American Journal of Pathology highlight includes an article titled MIR3783P Knockdown Recapitulates Many of the Features of Myelodysplastic Syndromes by first author Dahai Wang and Dr. Annette S. Kim. Myelodysplastic syndrome, or MDS, are a group of disorders caused by poorly formed or dysfunctional blood cells, generally associated with microRNA aberrant expression. MicroRNAs are small non-coding RNAs that bind imperfectly to regions of target messenger RNAs to repress their translation or production into proteins. The authors demonstrate that a stable knockdown of microRNA 3783P allows for an efficient model of aberrant MDS. Their model mimicked features of MDS, including altered immunophenotypic and morphologic dysplasia of white blood cells, and aberrant growth that characterized MDS with colony-forming assays, messenger RNA sequencing, and flow cytometry. The authors also found that chemotherapy drug azacitidine could reverse these malignant features from this microRNA loss. Check this article out online at the American Journal of Pathology website. Our article highlight from the Journal of Histochemistry and Cytochemistry comes from their December special issue on cancer stem cell niches. This article, titled The Histological Studies of the Ventricular and Subventricular Zone as Neural Stem Cell and Glioma Stem Cell Niche, by Asa Brockman from Dr. Rebecca Irie's lab. The summary provided by the authors states that the neural stem cell niche of the ventricular and subventricular zone supports the persistence of stem and progenitor cells in the mature brain. This niche has many notable cytoarchitectural features that affect the activity of stem cells and may also support the survival and growth of invading tumor cells. Histochemical studies of the niche have revealed many proteins that in combination can help to reveal stem-like cells in the normal or cancer context. Although many caveats persist in the quest to consistently identify these cells in the human brain. Here we explore the complex relationship between the persistent proliferative capacity of the neural stem cell niche and the malignant proliferation of brain tumors, with a special focus on histochemical identification of stem cells and stem-like tumor cells, an eye toward potential application of high-dimensional imaging approaches to the field. Check this article out to learn about neural imaging and the different methods of data acquisition to study the ventricular and subventricular zones of the neural stem cells.
Us today for Behind Our Science.